Hi, my name is Carly Martone, and I will be presenting genetic control of collective behavior in zebrafish. So the study uses zebrafish. There's a picture of some cute little zebrafish on the right there. Zebrafish are highly social. They swim in schools and exhibit a range of complex behaviors. So you might have heard of zebrafish being used in other genetic studies. This is pretty common, and zebrafish are good models because one, they're cheap. Two, zebrafish eggs are fertilized outside the body, so it's relatively easy to manipulate their genes without having to worry about actual reproductive aspects. And then three, zebrafish have a similar genetic structure to humans, making them a good model for human disease studies. In this study specifically, we already knew schooling behavior is under genetic control based on previous studies of zebrafish and a few other fish. We didn't know specifically which genes affected which behaviors, though. So, as I said, zebrafish are great models for human diseases. The bigger picture here is that the results of this study could be used to further understand social dysfunction in humans. We know conditions like autism and schizophrenia have significant genetic components, but this study could address specifically genetics related to changes in behavior that we see in humans with these psychiatric disorders. So the research question, which specific genes regulate collective behavior? This is a pretty broad question, but for one, no study previous to this one has been or had been done to the extent that this study had. And also, this study was really just aiming to identify which genes affect behavior, but not necessarily to determine if the genes that do affect behavior do so by altering sensory processes or like motor function. So this study kind of serves as a foundational study that would allow future studies to figure out some more specific types of processes that the genes would affect behavior using. So as for the methods, 90 different mutant lines were created using CRISPR-Cas9. That's a lot of zebrafish. You've probably heard of CRISPR-Cas9. It's used in many, many genetic studies, basically acting as scissors that cut DNA so then you can insert or delete genes. And that's how they created these mutant zebrafish. So for each mutant line, they took 3 females and 3 males, and they put them in a tank together, and they recorded their swimming behavior. So you can see that on the right here. Each trial they recorded for 30 minutes. They were looking specifically at the spacing in between the fish and also at the swimming patterns. So the video that's playing now is an example of normal zebrafish. So that's no mutations, just regular old zebrafish. The spacing is normal, and you can tell sometimes they swim together, but there's also some individual movements sometimes. So they took this video footage and they uploaded it, and they used a computer program to track each fish and essentially draw a line where they swam. You can see in this picture here, WT just stands for wild type. So this is again showing you an example of just a normal zebrafish. But then based on the photos of these results, they used other computer programs, MATLAB and Python, if you've ever heard of those. They use that to, one, account for the inaccuracy of the tracking program because it wasn't perfect, and also to put numbers to like quantify the spacing between the fish or where they swam in the tank, like patterns that way. So there were a ton of calculations that went into this study. So as for the results, they tested and analyzed all 90 genes and ended up grouping them into 3 different patterns. So some gene mutations resulted in a scattered pattern. You can see the 2 photo circles on the far right. They don't really show any pattern at all. So each of the fish kind of acts individually and does their own thing. There's not much group behavior between them. Other gene patterns resulted in other gene mutations resulted in essentially the opposite. So you can see in the top right photo circle, this is a coordinated pattern, and the fish here only followed each other and swam in a very fixed circular pattern around the outer part of the tank the whole time. Then there's a third group. So some gene mutations resulted in a huddled pattern. So you can see in the 2 photo circles on the left, these fish only swam in one specific part of the tank. There's no pattern really, but they did all cluster together just in one part of the tank. So what is the take-home message? The researchers did essentially accomplish their goal, and that was of identifying genes that regulate collective behavior. They explained that a limitation with this particular study is that they don't know what physiological process accompanies each gene mutation's effect on behavior. So they didn't analyze anatomical changes or neural activity. But that means this study creates a framework for future studies to analyze each gene more closely and determine how genes impact behavior, sensory processes, motor function, kind of look at each individual line in a more comprehensive way. So what's really fascinating is that some of the genes that have been previously linked to human psychiatric disorders, so a couple of the scattered pattern gene mutations are present in autism, and then some of the huddled gene mutation patterns are present in autism, schizophrenia, bipolar disease, and major depressive disorder. And then finally, the coordinated gene mutation behaviors have actually been seen in people that abuse nicotine and cannabis. So the hope would be that that a study like this one would eventually create a direction for treating these disorders, or at least the behavioral basis of them. So, if the zebrafish can model the disease themselves, hopefully they could model treatment options for humans with these types of disorders. That's all I got. So if you have any questions, feel free to email me. My email is carly@psu.edu. Thank you.